and welcome to the first episode of Bloodwork. I'm Dr. Sophie Callaway, an assistant professor from the Blood Cancer and Stem Cell Group at the University of Nottingham in the UK. I'm going to be using this channel to discuss the latest advancements in blood cancer research, things that are interesting, exciting, or just really important that I think you need to know about. I'm hoping that this will be an accessible channel for patients, families of patients, students who are looking to learn more about blood cancer, and also fellow researchers. My own research focuses on acute myeloid leukemia, so I'll be starting here, but I will be expanding into other blood cancers as time goes by. And I'm going to cover a variety of topics, so it might be that latest paper that everyone needs to know about, but I will also be covering some broader topics. And today I'm going to be focusing on UBTF tandem duplication AML as one of these broader topics. Now, you might be wondering what on earth a UBTF tandem duplication is, as whether you're a patient or a genomics geek like me, they're not very well known. In fact, we've only known that this type of mutation causes AML and myelodysplastic syndrome for around about 3 years. And it is fairly rare compared to more well-known mutations like FLT3-ITD you might have heard of, with only a handful of cases for every 100 patients diagnosed. Now, tandem duplications happen when part of the gene is copied more than once, disrupting how the protein functions. So this mutation happens when part of the UBTF gene, which rather wordily stands for upstream binding transcription factor, is duplicated. Just like this. Now, in a healthy blood cell, UBTF is a key player in making the machinery necessary for the cell to function properly. And with such an important role, it may come as no surprise that UBTF tandem duplication AML is associated with a really poor prognosis, with an overall survival of less than 2 years on average. And even worse, UBTF tandem duplication is most often found in young adults and children. And one specific problem with this type of AML is that it seems to really lead to problems with relapse. With only a little over half of UBTF mutated patients actually achieving complete remission from standard chemotherapy, this is a huge concern for our younger patients. So let's dive into what is being done to help solve these challenges, because there have been some really interesting and rapid advances in understanding this type of AML. So just a couple of months ago now, a team from Barcelona were able to show that measurable residual disease monitoring really effectively predicts which UBTF tandem duplication patients would go on to relapse. So measurable residual disease monitoring, or you might have heard of it as minimal or molecular residual disease, is a process of tracking whether any cells remain after treatment, whether any AML cells in particular remain after treatment. And this helps select those patients who really need different treatments or more treatments. And in this paper, the group could do this by measuring the expression levels of a gene called WT1, or in other words, how much WT1 the cells are trying to make. And WT1 is often overexpressed in AML compared to healthy cells, and it's usually most highly expressed in the very immature leukemia-initiating cells, and these are the ones which go on to cause relapse. So measuring the expression of WT1 has been used before for measurable disease monitoring for some time now, with mixed success. But it does seem to really work well as a marker for residual disease in UBTF AML. Now, the caveat here is that there were only small numbers of patients in this study, and it is going to need to be expanded to more groups. So only 2 of the patients in this study did go on to relapse. But I think this is really promising. So it's great that we can predict which patients may relapse, but it means that we still need better and more treatment. So what treatment options are we looking at here for these types of patients? Well, this brings me on to the other important side of UBTF tandem duplication AML, which is that these cells express HOX genes. Now, HOX genes are a really important cluster of genes which provide instructions for making mature blood cells out of stem cells, and in fact lots of mature cells out of stem cells. And they're so crucial that you see them right the way from flies up to humans in all parts of the body. And Hox gene expression is also an important way in which certain AML cells are kept as the cancerous stem-like cells and blocked from becoming healthy, functional blood cells that we actually want. So a lot of research over the past few years has focused on disrupting Hox gene expression in AML. And one of the most important advancements in this is menin inhibitors, which you may have heard of. They are currently in clinical trials with MLL-rearranged leukemias in particular. And menin inhibitors act to damp down Hox gene expression by disrupting the complex between the mutated MLL and menin, and together these activate Hox expression. And these proteins have to sit together on the DNA in order to activate Hox genes. So what about the patients with UBTF tandem duplication AML? Well, a joint endeavour by teams from St Jude's and the Princess Maxima Institute showed that in UBTF tandem duplication AML, the Hox genes are expressed because the mutated UBTF also sits with MLL and menin on the DNA at the Hox genes. And as a result, this meant that the UBTF AML, like MLL rearranged AML, is sensitive to the menin inhibitor revumenib, the one that's currently in the clinical trials. So together, these two studies offer a very interesting possibility for these patients. One, we could identify the patients with UBTF tandem duplication AML who have responded poorly to chemotherapy. They're maintaining a population of stem-like cells able to cause relapse. And two, we could then maybe try revumenib with them. Maybe this would be more curative in these patients because it's also targeting these most stem-like cells that we're actually identifying by measuring the residual disease with WT1 expression. And menin inhibitors have shown great promise in clinical trials so far, particularly where chemotherapy has failed. And I'm not the only one who thinks so, which is great, because last summer a letter in the New England Journal of Medicine reported a case of the compassionate use of revumenib in a relapsed post-transplant UBTF patient. And in this case, revumenib managed to give the patient a further 5 months in remission, although unfortunately after this, resistance did once again occur. But they're working on the next generation of these drugs already, so watch this space, and hopefully this will be a real great advancement for these patients who are really not doing so well. So I hope that you found this interesting. I'm going to be aiming to release a new episode each month looking at the important advances in blood cancer research. So if you're interested in staying up to date with the latest breakthroughs, make sure to subscribe, and I'd love to hear your thoughts or any questions that you'd like me to address in future episodes in the comment section.